हेलो बच्चों, वेलकम टू SPSVERSITY. In the previous session, we discussed about the glycolysis and the end products. And in the presence of oxygen, it will proceed for aerobic respiration. And in the absence of anaerobic respiration, it will proceed for fermentation. इन दिस सेशन हमें डिस्कस करेंगे एरोबिक रेस्पिरेशन के बाद. सो ग्लाइकोलाइसिस के बाद में पायरुवेट इज फॉर्म्ड इन साइटोप्लाज्म. So this pyruvate should be transported to mitochondria. So the first is the pyruvate should be transported to mitochondria. In mitochondria, we can see the complete oxidation of pyruvate stepwise. Where after oxidation we can see electron transport also, where oxygen is the final acceptor of hydrogens and electrons for the synthesis of ATP. पहले हमें देखेंगे पायरुवेट को क्या होगा. What is the fate of pyruvate? So this pyruvate undergoes three reactions. पहला reaction oxidation, decarboxylation, और next reaction condensation. ऑक्सीडेशन, डिकार्बोक्सिलेशन, कंडेंसेशन के लिए एक एंजाइम चाहिए. इट इज पायरुवेट डिहाइड्रोजनेज कॉम्प्लेक्स. इसमें Mg2+ लिपोइक एसिड, थायमिन पायरोफॉस्फेट, NAD को एंजाइम A रहता. So these 2 moles of pyruvic acid, in the presence of pyruvate dehydrogenase complex, it will undergo oxidation, decarboxylation, condensation. So finally what we get by this is 2 moles of acetyl coenzyme A and 2 carbon dioxide along with 2 NADH2. So by these 3 reactions, by the end we'll get 2 moles of acetyl coenzyme A, 2 molecules of CO2 and 2 NADH2. NADH2 which is energy currency, 2 CO2 which is released in the atmosphere. And these 2 acetyl coenzyme A will proceed for Krebs cycle. Krebs cycle और दूसरे नाम है ट्राइकार्बोक्सिलिक एसिड साइकिल, और इसको क्या बोलते हैं कि सिट्रिक एसिड साइकिल. So Krebs cycle, tricarboxylic acid cycle, citric acid cycle, or same. So this Krebs cycle takes place in the matrix of mitochondria. So all the enzymes, reactants which are required for this Krebs cycle are present in matrix of mitochondria. Except succinate dehydrogenase, which is located in inner membrane of mitochondria. So पूरा enzymes मैट्रिक्स ऑफ माइटोकॉन्ड्रिया में रहता है. सिर्फ एक ही एंजाइम सक्सिनेट डिहाइड्रोजनेस इनर माइटोकॉन्ड्रियल मेम्ब्रेन में रहता है. एक क्रेब्स साइकिल में for one acetyl coenzyme A, 3NADH2, 1 FADH2 and 1 ATP is produced. So, एक एसिटाइल कोएंजाइम के वास्ते क्रेब्स साइकिल में 3NADH2, 1 FADH2 and 1 ATP प्रोड्यूस होता है. It is a cyclic process. It's a cyclic process. So देख सकते हैं, a cycle of reactions will take place in cyclic manner. Whereas glycolysis is a linear one. Whereas citric acid cycle is a cyclic process. इसमें step wise देखेंगे, step 1, step 2, step 3. पहला स्टेप 1 में एसिटाइल कोएंजाइम it condenses with oxaloacetate. So oxaloacetate is a 4-carbon compound. Acetyl coenzyme is a 2-carbon compound. On condensation, it will form a 6-carbon compound. इसका नाम citrate or citric acid. In the presence of enzyme citrate synthetase, this is a पहला reaction. This citrate synthetase, the next reaction is the second one. It undergoes dehydration. So dehydration means removal of water. In the presence of enzyme aconitase, a cis-aconitate formed. This cis-aconitate, in the presence of aconitase, it undergoes hydration. हाइड्रेशन के वास्ते क्या फॉर्म होता है कि आइसोसिट्रेट, it will form isocitrate. This is reaction number 3. This isocitrate will undergo 2 reactions. One is oxidation and another one is decarboxylation. Oxidation means release of H2 ions. Decarboxylation means removal of CO2. In the presence of enzyme isocitrate dehydrogenase. इसके वास्ते क्या फॉर्म होता है कि alpha ketoglutarate, which is a 5-carbon compound, which is a 5-carbon compound. So this alpha ketoglutarate, which is a 5-carbon compound formed from isocitrate, it also undergoes oxidation, removal of H2, and it undergoes decarboxylation. So due to oxidation, it will form NADH2, and due to decarboxylation, it will release CO2. It will release CO2. And it will form a 4-carbon compound succinyl coenzyme A. It will form a 4-carbon compound succinyl coenzyme A. So this succinyl coenzyme A will undergo splitting. So on splitting, it will form succinic acid and coenzyme A. Along with ATP is released. So succinate, which is a 4-carbon compound. So this succinate on oxidation, it undergoes oxidation. The oxidation की वास्ते it will form FADH2 and fumarate. Fumarate on hydration, that is addition of water, it will form malate. So it will form malate. Malate is also a 4-carbon compound. This malate will undergo oxidation. So इसके वास्ते इधर NADH2 फॉर्म होता है और oxaloacetate form. So finally again we'll get oxaloacetate. If you see this, all the reactions, it is for only 1 mole of acetyl coenzyme A. So this 1 mole of acetyl coenzyme A on Krebs cycle, so it will form 3NADH2. If you look into the cycle, देख सकते NADH2 किधर फॉर्म हो गया, इधर एक NADH2, इधर एक NADH2, and the last reaction इसमें एक NADH2. 1 FADH2 between succinate to fumarate, 1 FADH2, or 1 GTP from alpha succinyl coenzyme A to succinate. This is the final output of energy from Krebs cycle. और इसके साथ 2 CO2 molecules are released. इसके साथ 2 CO2 molecules are released. This is for 1 acetyl coenzyme A. If we take for 1 glucose, एक ग्लूकोज से ग्लाइकोलिसिस के बाद में 2 पायरुवेट आता है और 2 एसिटाइल कोएंजाइम. For 1 glucose, this cycle will take place for twice. The same cycle takes for twice. So everything becomes double. That is 6 NADH2, 2 FADH2, 2 ATP or 4 CO2. So for 1 glucose, when participates in, that is in Krebs cycle, if we take 6 NADH2, 2 FADH2, 2 ATP, 4 CO2 are released. If you look into the reactions of biochemical reactions step by step, this is step 1, that is acetyl coenzyme A on condensation with oxaloacetate will form citric acid. The first stable compound is citric acid. That's why it is called citric acid cycle. और सिट्रिक एसिड साइकिल में 3 कार्बोक्सिलिक एसिड रहता है, इसलिए इसको दूसरा नाम है कि ट्राइकार्बोक्सिलिक एसिड साइकिल. This citric acid will undergo dehydration and form cis-aconitic acid. Cis-aconitic acid, on hydration, it will form isocitric acid. Isocitric acid on oxidation and decarboxylation, it will form alpha ketoglutaric acid and NADH2. Okay. So this alpha oxalosuccinic acid will undergo decarboxylation and form a 5-carbon compound and release CO2. So alpha ketoglutarate, in the presence of dehydrogenase, alpha ketoglutaric dehydrogenase complex, it will undergo oxidation, decarboxylation and condensation, and it will form a 5-carbon compound, succinyl coenzyme A, along with CO2 and NADH2. Succinyl coenzyme A will undergo splitting in the presence of thiokinase. This means succinic acid, CoA is released along with ATP. 1 ATP is released. Succinic acid in the presence of succinic dehydrogenase will undergo oxidation and release fumarates. Fumarate and hydration, it will form malic acid. Malic acid and oxidation, again it will form oxaloacetic acid, which is the acceptor of acetyl coenzyme A. So by the end, from 1 acetyl coenzyme A, 3 NADH2, 1 FADH2, 1 ATP, 2 CO2 और release. So इधर देख सकते हो. For 1 acetyl coenzyme A, it is 3, 1 ATP, 3 NADH2, 1 ATP, 1 FADH2. For 1 glucose means 2 acetyl coenzyme A, the Krebs cycle will take place for twice. So twice means everything becomes double. 6 NADH2, 2 ATP, 2 FADH2. So finally, if you look into this, this is the energy output from Krebs cycle. Okay. So this Krebs cycle takes place in matrix of mitochondria. बाद में क्या होता है? So बाद में हमें देख सकते electron transport. We look into differences between glycolysis and Krebs cycle. Okay. Glycolysis is common step for aerobic as well as anaerobic, but whereas Krebs cycle is seen only in aerobic respiration. Glycolysis cytoplasm में होता है, लेकिन Krebs cycle matrix of mitochondria. ग्लाइकोलाइसिस एक लीनियर पाथवे है, एक साइक्लिक पाथवे. ग्लाइकोलाइसिस से ग्लूकोज से पायरुवेट आता है, इधर से एसिटाइल कोएंजाइम. इसमें 2 एटीपी कंज्यूम होता है, इधर नो कंज्यूम, एटीपी कंज्यूमिंग नहीं रहता. इसमें 2 एटीपी रिलीज होता है, इसमें 2 ATP रिलीज होता है. इसमें 2 NADH2, इसमें 6 NADH2, 2 FADH2 और 4. इसमें CO2 नहीं रिलीज होता है, लेकिन इसमें CO2 रिलीज होता है. So this is the differences between glycolysis and Krebs cycle. So Krebs cycle के बाद में aerobic respiration में step है कि electron transport system which occurs in inner mitochondrial membrane. So electron transport system, here the name itself says there is a transport of electrons. So what actually happens in electron transport system? इसमें सिर्फ NADH2 and FADH2 are converted to ATP. इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में क्या होता है कि सिर्फ इसमें कन्वर्शन ऑफ NADH2 to and FADH2 to ATP. So they should be converted to ATP. Then only they can be used for various process taking place inside the cell. इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम इसमें क्या रहता है? इसमें सीरीज ऑफ अरेंज्ड 9 इलेक्ट्रॉन कैरियर्स रहते, उसको क्या बोलते? कोएंजाइम्स। वो 9 है कि NAD निकोटिनामाइड एडेनाइन डाइन्यूक्लियोटाइड, फ्लाविन मोनोन्यूक्लियोटाइड FMN, फ्लाविन एडेनाइन डाइन्यूक्लियोटाइड FAD, कोएंजाइम Q, साइटोक्रोम B/C1, साइटोक्रोम C, cytochrome A3, 9 electron carriers. And these 9 electron carriers are organized in the form of complexes. इसमें इलेक्ट्रॉन ट्रांसपोर्ट चेन में 4 प्रोटीन कॉम्प्लेक्सेस और 2 मोबाइल कैरियर्स रहते। 4 प्रोटीन कॉम्प्लेक्सेस और 2 मोबाइल कैरियर्स। इसमें कॉम्प्लेक्स 1 लिए तो इसमें NADH डिहाइड्रोजनेस रहता। कॉम्प्लेक्स 2 में सक्सिनिक डिहाइड्रोजनेस रहता और कॉम्प्लेक्स 2 और 3 के बीच में एक मोबाइल कैरियर रहता, वो उसको क्या बोलते हैं कि कोएंजाइम Q और ubiquinone EQ। कॉम्प्लेक्स 3 में साइटोक्रोम B और C रहता, which is associated with non-heme iron। कॉम्प्लेक्स 4 में साइटोक्रोम A और A3 रहता, which is a copper bound one। इसमें इलेक्ट्रॉन्स विल फॉलो ए पाथवे की इलेक्ट्रॉन्स कॉम्प्लेक्स 1 से शुरू होकर 3, 4 ए पाथवे और दूसरा पाथवे की 2, 3, 4 कॉम्प्लेक्सेस। देखेंगे कॉम्प्लेक्स 1 से क्या शुरू होता है, कॉम्प्लेक्स 2 से क्या शुरू होता है। इसमें NADH2 which is formed here, it will start from complex 1. FADH2 which is formed in Krebs cycle, it will start from complex 2. याद रखना है कि NADH2 in ETC will start from complex 1. FADH2 will start from complex 2. NADH2 will undergo oxidation and release electrons which are taken by complex 1. The same process will take place. FADH2 will undergo oxidation and release electrons which are taken by complex 2. So complex 1 or 2C, electrons through mobile electron carrier that is coenzyme Q, it is taken through complex 3. कॉम्प्लेक्स 3 से अगेन कॉम्प्लेक्स 3 और कॉम्प्लेक्स 4 के बीच में साइटोक्रोम C रहता. So this cytochromes will C carry electrons from complex 3 to complex 4. Complex 4 में cytochrome A and A3 रहते, which contain 2 copper centers. Okay. As electrons will move from to different protein complexes and mobile carriers. Okay. We'll look into the diagram very clearly. So this is inner mitochondrial membrane. A outer mitochondrial membrane और inner mitochondrial membrane के बीच में intermembrane space, उसको क्या बोलते हैं कि intermembrane space. और inner mitochondrial membrane के नीचे matrix of mitochondria है. A protein complexes A inner mitochondrial membrane में रहता और a protein complex के साथ mobile carriers भी mitochondrial membrane में रहता. A complex 1 है. A complex 2, a mobile carrier quinone, a complex 3, complex 4, complex 5. और complex 3 और 4 के बीच में साइटोक्रोम C मोबाइल कैरियर रहता. Either complex 1 say NADH2 will start its journey. NADH2 on oxidation, it will release electrons which are taken by complex 1. And again, these electrons will taken by quinone which will carry to complex 3. Complex 3 say mobile carrier C, that is cytochrome C, will take these electrons and transfer it to complex 4. A complex 4, finally electrons will reach to complex 4, and from complex 4 the electrons are accepted by oxygen along with 2H plus and it will form water. So this is the pathway of electrons of NADH2. Complex 1, quinone, complex 3, cytochrome C, complex 4. Final terminal acceptor of electrons is oxygen. Along with electrons, it will take 2 protons and it will form water. FADH2 लिए तो FADH2 will start its journey from complex 2. A complex 2 से शुरू होके electrons will be moved to quinone. Quinone से complex 3. Complex 3 से cytochrome C. Cytochrome C से complex 4. Finally this. So this is the flow of electrons, that is when NADH2 participates in electron transfer. And FADH2, this is the flow, and for NADH2, this is the flow of electrons. Okay. So during this electron transport process, अब पिक्चर में देख सकते हैं, protons are transferred to matrix to intermembrane space. So matrix से intermembrane space को protons are transferred. So these protons are transferred from matrix to intermembrane space. इसके लिए क्या होता है? So simultaneously during electron transport, protons are transported from matrix to intermembrane space. Due to this, what happens is intermembrane space becomes with high concentration of protein, जिसमें high concentration of protons रहता है. मैट्रिक्स में लो concentration of protons होता है. इधर ज्यादा रहता, इधर कम रहता. सो इसको क्या बोलते कि प्रोटॉन ग्रेडियंट. इसको क्या बोलते? प्रोटॉन ग्रेडियंट. A proton gradient के वास्ते a force is created, that is proton motive force. A proton motive force बोलते क्या? That is the movement of protons from high to low concentration. So protons has to move from intermembrane space to matrix. लेकिन intermembrane space, it will not allow these protons. So protons has to move through complex 5. इसको क्या बोलते कि ATP synthase. इधर देख सकते हो, direction of protons is like this, that is from intermembrane space to matrix. Here protons are moving from high concentration to low concentration. So इसके वास्ते क्या होता? As protons are moving from high to low concentration, energy is released. This energy is used for combining with ADP with inorganic phosphate, and it will form ATP. It will form ATP. So finally, NADH2 and FADH2 are converted to ATP. इसमें 1 NADH2 is equal to 3 ATP. From 1 NADH2, 3 ATP are formed. And from 1 FADH2, 2 ATP are formed. This is the final conversion of NADH2 and FADH2 to ATP. So finally it is formed. So by the end of this electron transport chain, NADH2 and FADH2 are converted to ATP. So here we'll discuss about ATP synthase, which is called complex 5. Which major function is ATP synthesis? इसमें दो components रहते, F1, F0. F1 is head piece, which is peripheral protein complex, where protein ATP synthesis होता. F0 is an integral membrane protein, which is like a channel where protons will pass through. So diagram देख सकते. This is F0, which is just like a channel. This is F1, which is called head piece, where ATP synthesis होता है. So this is what finally happens in electron transport chain. And if we convert into ATP, each and every step, that is if we take in glycolysis, 2 ATP are formed, or 2 NADH. Each ATP, each NADH to 3 ATP, 6 ATP. Formation of acyl coenzyme from pyruvate, no ATP, only 2 NADH2. Multiplied with 3, 6. Krebs cycle में 6 NADH2 or 2 FADH2 or 2 ATP. 6 multiplied with 3, 18 ATP. 2 FADST, it will form 2 ATP, finally you will get 4 ATP. That is total. If you take total net gain of ATP, it is 38 ATP. Net gain of ATP is 38 ATP in aerobic respiration. So aerobic respiration, say, finally we'll get 38 ATP, which is very higher than when compared to anaerobic respiration. This is generally what happens in respiration. Next हम देखेंगे glyoxylate cycle, which is discovered by Kornberg during germination of fatty seeds. इसमें क्या होता है? So during germination of seed, fats are converted into sugars. एक pathway, gluconeogenesis. And this occurs in glycosome, cytosol and mitochondria. और हमें respiratory balance sheet के बारे में लेते हैं. तो finally we get 36 ATP in aerobic respiration from 1 molecule of glucose. Finally get that. और respiration में हमें क्या पढ़ा है? कुछ intermediate substances. So where respiration is a catabolic process. लेकिन depending upon the need, some intermediate substances like alpha ketoglutarate, okay, and some are fatty acid precursors. So depending upon the need, they may, instead of going to the process of respiration, they may go for synthesis of amino acids or fatty acids, which is an anabolic process. So some intermediate substances like alpha ketoglutarate, which is a precursor of amino acids, and some are precursor of fatty acids. Instead of proceeding to respiration, a catabolic process, it will may proceed to anabolic process. इसमें सिर्फ catabolic नहीं, anabolic process में भी proceed कर सकते. इसमें दोनों रहते हैं, catabolic और anabolic दोनों रहते. So इसको क्या बोलते हैं कि amphibolic. Amphi means both. Here both means catabolic and anabolic process. हमें सिर्फ पढ़े अभी तक ग्लूकोज पार्टिसिपेट किया तो क्या होता, लेकिन सिर्फ कार्बोहाइड्रेट्स नहीं, प्रोटीन्स और फैट्स भी रेस्पिरेटरी सब्सटेंस. Proteins, if they participate in respiration, what is the process? Fats, if they participate in aerobic respiration, from where they will start? If you take proteins, proteins are simplified to amino acids. And these amino acids will form pyruvic acid. A pyruvic acid से शुरू होता है, a glycolysis नहीं रहता. So proteins, when involved in respiration, there will be no glycolysis. It will start from only pyruvic acid. Fats are simplified to fatty acids and glycerol. So glycerol will form, that is, it will form into dihydroxyacetone phosphate. It will start from dihydroxyacetone phosphate. Fatty acids will start its journey. It will be transformed into acetyl coenzyme E. So it will start its journey from acetyl coenzyme E. So these are different respiratory substrates when they are involved in aerobic respiration. Okay. Next concept is respiratory coefficient. So respiratory quotient polatu volume of CO2 evolved by volume of O2 absorbed. Okay. To measure this, we require Genong's respirometer. Carbohydrates लिए तो इसमें 6 O2 are used, 6 CO2 are released. So 6 divided by 6, 1. Fats और oils, fats में ज़्यादा O2 utilize होता, less CO2. That is less means 0.7. Organic acids mean more CO2 is released and less oxygen is taken. 4 by 3, that is more than 1. Oxalic acid का RQ value 4 रहता और citric acid RQ value 1.3. Proteins में है तो it is also less than 1, 0.8 or 0.9. Anaerobic respiration में सिर्फ CO2 रिलीज होता, O2 नहीं यूज होता. 2 divided by 0, which is infinity. So by seeing the RQ value, we can see what respiratory substrate it is involved. Okay. And when you come to factors affecting the rate of respiration, lots are factors. Temperature, oxygen, CO2, water, like this. Temperature, which is that is 20 to 35 degrees optimum temperature, where enzymes are active at this temperature. If the temperature is high, what will happen? Enzymes get denatured, they will not function. If the enzymes are kept at low temperature, inactive due to inactivation. Okay. So optimum temperature for respiration is 20 to 35 degrees, maximum is 45 degrees. Oxygen, inhibition of anaerobic respiration is by increase of O2 is called Pasteur's effect. If CO2 concentration increases, the rate of respiration in plants will decrease. Why? Because it leads to closing of stomata. CO2 concentration ज़्यादा हो गया तो स्टोमेटा क्लोज होता है. Salts हमें लिए तो. So if a plant is transferred from water to salt solution, क्या होता है? Respiratory rate increase होता है. इसको क्या बोलते हैं कि salt respiration. Why it will increase? Because absorption of ions requires more energy. कुछ hormones they will increase the respiratory rate like IAA, indolistic acid, gibberellic acid. Cytokinins will increase the respiratory rate. Okay. And even during ripening of fruits and senescence of leaves, even respiration will increase. इसको क्या बोलते हैं कि climacteric respiration. Okay. Later on it will decrease due to production of ethylene. Light also will influence the respiratory rate. Due to light, it will influence opening of stomata, which will increase the respiration. Injury or any damage or diseases, wounds also will increase the respiration. This is required for healing process. Age, young cells में respiration rate बहुत ज़्यादा रहता है. Like in meristums, you can see respiratory rate is more when you compare with permanent tissues. So this is all about the respiration in plants, where we discussed about aerobic respiration and anaerobic respiration. Thank you all. Follow us on social media, SPSVERSITY. We'll meet again in the next session.